much of this work has been inspired by medical science, but it has very significant implications and opportunities in animal science and in conservation. The organisation that I've been involved with is a Texas-based organisation known as Colossal Biosciences that is owned and bankrolled by the man on the right here, Ben Lamb, who is a billionaire who has made his fortune in biotech and in AI technology. And he's based in Texas, Fort Worth, in a large facility there with over 270 scientists. The head of science is the lady on the left there, Beth Shapiro, Dr. Beth Shapiro, who is a leader in the technology of especially ancient DNA. She worked with Professor Alan Cooper at Oxford University. Alan Cooper's a New Zealander. This group were the first to produce the complete genome of the dodo, Beth was the lead author on the work that identified that beyond any doubt the dodo was in fact a very large flightless pigeon. That's a dodo in the background behind. It's not quite as large as that. It's about the size of a turkey, but it's a very strange looking animal, and for more than 150 years the exact relationships of the dodo to other birds was poorly understood. But now Colossal is very keen on bringing back the dodo as part of their first avian de-extinction. But the name Colossal, of course, actually comes from the mammoth, which is a species that Beth, about 15 years ago, wrote a book about how to de-extinct the mammoth. Ben read this book, understood the technology to the point that he understood that it would definitely be possible to de-extinct a mammoth. And the mammoth has been the poster child of this company and led to the name Colossal, of course, the largest land mammal. The process of de-extincting a mammoth is not simple. They have an extremely long gestation time, but they've made considerable progress at that. Some of you may have seen the fact that they were able to engineer mice which actually had the hair of mammoths last year. But their first major success was the dire wolf, a species of extremely large wolf that became extinct about 10,000 years ago during the last glacial maximum, and a species that was it was in popular culture. It appeared in the Game of Thrones series. When this was announced in April, it led to a little bit of controversy in the fact that the definition of exactly what de-extinction was questioned. What we're going to be talking about here today is functional de-extinction, the fact that you can create, using the genes of the animals themselves, animals which, to all intents and purposes, talk like a duck, quack like a duck, and therefore are a duck. The idea of complete replacement of a living animal's cells with the complete genome of an extinct animal is, and perhaps always will be, beyond our reach. But what we can, beyond any doubt, do is create an animal that, to all intents and purposes, is that looking like a duck and quacking like a duck animal. So another project other than woolly mammoth that is underway with Colossal, in association with the University of Melbourne and the Indigenous people of Tasmania, is a project to create a thylacine. This had a major breakthrough last year when the complete genome, what they call the telomere to telomere genome of thylacine, was actually produced. And now we're in the process of, and Dr. Andrew Pask at the University of Melbourne, who's a full-time employee of Colossal, is in the process of bringing the thylacine equivalent back to life and introducing it into its original home in Tasmania. Another side of Colossal's business is actually the creation of biovaults. So just last week there was an announcement that in the United Arab Emirates, a large facility, one of several around the world, including one that will be in New Zealand, has been funded, and this will have the ability to store the germ tissue of up to 10,000 different species in these large cryogenic vaults. These vaults obviously will have the redundancy of the fact that there will be vaults all around the world. And these vaults aren't just this storage facility due to the fact that everything may or may not become extinct, but they also provide the ability for us to have viable tissue stored cryogenically that can be then used in conservation projects. For example, in species in places where conservation isn't currently possible, war zones, places in the third world. At the very least, we can actually just store viable germ tissue of these animals, and so using the technologies that we're developing with regards to each of the different animal groups, mammoths, birds, and as I'll be talking about moa, this should mean that in the future any unfortunate extinctions we can actually reverse. And secondly, we should also be able to have tissue available for populations that are in severe decline that will enable us to perform what's known as a genetic rescue by placing the individuals with genetically different information back into a severely reduced population. But I'm just going to take a step forward, or sideways, for a second, and talk about Aotearoa New Zealand and the moa itself. So when Polynesian Maori first arrived in New Zealand, they found a remarkable landscape, a remarkable ecosystem that was unlike anywhere else on Earth. An ecosystem that didn't have any terrestrial mammals, an ecosystem that was ruled by these giant birds. And although our understanding of early Polynesian arrival is informed by the by Maori mythology and lore. The full understanding of exactly how this happened has only quite recently been accepted by, I guess you'd say, Pākehā science. And our understanding that these Polynesian arrivals weren't these lost people, but a group of migrants, highly skilled navigators coming to Aotearoa, New Zealand, and going backwards and forwards across the seas has really developed over the last only 50 or 60 years. And, you know, the idea of these large waka that are similar to the one that's just recently been found on the Chatham Islands, the Kohu, has really gave us an understanding of exactly how and why the landscape in New Zealand was quite dramatically affected by people's arrival. So when Māori first arrived, they found a land with these giant birds in. The interesting part of this story is the fact that Moa is the name that these birds were given, or one of the names that these birds were given. Elsewhere in Polynesia, moa is actually the word for chicken. There were, in fact, 9 different species of moa. This illustration shows 7 of those, and they were very different in size and in habits. The largest, of course, were the giant moa. There were 2 species of those, one in the North Island and one in the South Island. And what we've been able to do over the last 20 or 30 years is actually nail down exactly how many species of moa there were and exactly how these different species were related. If you go back to the era prior to genetic studies, you'd find in old textbooks information which goes, suggests that there could have been as many as 38 different species of moa. Even when I first became involved, there were 11 or 12 species known, but by refining, using techniques mainly related to DNA, we've now come to a reasonably firm conclusion that there are 9 species that were alive when humans first arrived in New Zealand. And they belong to 3 different families. The Maids, which include, I guess, probably the most numerous of the moa, especially Emmaus and Euryaptrix, which were the 2 species that were most abundant, for example, in the Canterbury region. The Deinonythids, the giant moa, just contained these 2 different species, and then there was the Megalaptrid, the upland moa, which was only found in the South Island, in the upland areas of the South Island. And the 9 species that we have were all had a single common ancestor that we believe was only about 6 million years old. So this means genetically these animals are very closely related. By comparison with other groups, say for example birds, where the splits from some of the bird species go back 66, 70 million years. This doesn't mean that prior to 6 million years ago there was only a single species of moa in New Zealand, and some of the work that I'm doing currently at St Bathans in central Otago has actually shown that we have a number of different species of moa from that era, 20 million years ago, and that what the DNA story that we've we've finalised actually tells us is that the moa as a group in New Zealand went through what's known as a genetic bottleneck about 6 million years ago, which coincides with the beginning of the ice ages, and suggests that only a single common ancestor actually made its way through the catastrophic period of the ice ages and led to the diversity of moa species that we have in New Zealand today. So we'll just recap this moa diversity. We have the giant moa, one species in the North Island, one species in the South Island. Remarkable for its sheer size, had one of the highest reaches of any bird in the world, weighed 250 odd kilos, a very large female, and had a very, what we call a high sexual dimorphism. But this was what's known as a reverse sexual dimorphism, so the female was 2/3 the size of the male. The heavy-footed moa had 3 species, and as you can imagine, being called heavy-footed, it had this extremely heavy foot, and the North Island species was considerably smaller, but these were sort of like, what would you call it, wrecking balls, or they actually remind me a lot of the wombats, which you may or may not be familiar with in Australia. Heavily, very unusually heavy for their size or for their height. And you know, if one of these was running down a path towards you, you definitely want to get out of the way. Then the 2 species I was mentioning before that were common in Canterbury, Euryaptrix, sometimes called the stout-legged moa. Not very big fan of the common names of these. I mean, I can never remember if it's heavy or stout, and so I generally just refer to them by their scientific names. And then the coastal or eastern moa, Emmaus, also one of the more common species in the Canterbury region. The little bush moa, obviously found primarily where there was forest, not so abundant on the Canterbury Plains, and you know, very common, for example, their remains are very common in the cave systems of the North and the South Island, also quite abundant in Southland. And the upland moa, which I alluded to before, comparatively small species, only a little bit bigger than a turkey, and also a species that had quite a remarkable range. It seems to have been primarily in the area above the tree line in the South Island along the Southern Alps. Probably came down into the forest during the winter, or definitely came down into the forest during the winter, otherwise it wouldn't have been able to forage. And this is actually quite similar to a couple of other species that were restricted to the upland areas in the South Island. This is the species that evidence suggests was probably the most long-lived of the species. It became extinct the latest, maybe 1500. And that's most likely because of the fact that it avoided the factors that led to the extinction of each of the moa species, which were a combination of moa hunting, the loss of habitat, the effect of kuri, the Maori dog, that were abundant and wild in the landscape, and possibly also disease. Now, although we don't have the full picture about moa, the work of a number of specialists around the country, most particularly Jamie Wood, who was at Landcare Research, have over the last 20 odd years led to our far better understanding of moa biology. We now have these coprolites, these just mummified poo mostly from Southland and a couple of sites in Otago. And they just give us a glimpse into our understanding of the biology of these animals. Clearly, because of their size and their abundance, they had a very important role in the ecosystem. But up until the work on DNA was done, we only really had some very cursory ideas of what the effect was. And there was a suggestion that given their abundance and size and the fact that they were herbivorous, that they may have had a very similar effect on the ecosystem as the introduced ungulates, especially the goats and the deer, which was quite a controversial idea. And given the fact that New Zealand forests are so dense, the idea that in their natural state, the idea that moa may have actually contributed to the opening up of the New Zealand forest was quite a significant idea. But the bottom line is that the work that has been done on their diet suggests that remarkably the way that they forage was fundamentally different from the way that deer forage, and that whilst unquestionably the forest canopy would have over a long period of time be affected by this browsing, and the understory was also affected. It was not affected in the same ways that moa affected the understory. And therefore there is no equivalent to creating a New Zealand forest using ungulates rather than moa. There are a couple of other interesting stories about moa that have really significant roles in our understanding of the animals or the flora that we see in New Zealand today. One is the story of divaricating shrubs. You'll be very familiar with these. Many of you will have these in your gardens. These plants which have sticks on the outside and leaves on the inside. I'm not going to go into what I believe is the correct hypothesis for this, but two competing hypotheses. One is that this evolved to avoid moa browsing. Another is that this evolved to avoid hoarfrost and the effects of these very severe frosts by actually putting ice on the outside and then the ice not affecting the inside. Those are two competing ideas. Today is a forest which is literally virtually impenetrable. I've been to the Amazon rainforest, the jungles of Asia, and frankly they've got nothing on a west coast forest today. You can get through any sort of jungle pretty easily, but to get through the back country behind Punakaiki when you're off the track is truly an impenetrable forest. And we know for a fact that moa were found in caves in these forests, that they'd fall into their deaths in these caves. A moa, I can tell you right now, couldn't have got through the forests that are there today, and the only explanation is that moa over millennia had fundamentally changed the shape of a New Zealand forest. Similarly, in the landscapes, especially of Canterbury, where we have these large flats here with forest on either side, this structure, the fact that there's grassland on either side is sometimes called a frost flat. And we know that moa were quite abundant in this sort of landscape, and they would have had a very significant role in creating this landscape. These sort of frost flat landscapes we know were very important habitat for a number of species that have become extinct. Open land species such as the takahe, or almost extinct, or the adzebill. And yet these sort of habitats are firstly highly grazed now by sheep and cattle, but in places where they're not being highly grazed, there's major problems with willow and pine taking over these areas. And effectively what is unfortunately the case is that there's no place even for some of these species such as takahe. And kakapo was another species that was very common in this sort of landscape. And then we have interesting aspects of erosion. There's some people that see crisscross tracks up these heavily sheep-grazed environments, and suggest that those are actually caused and were caused by moa. So all in all, we have an understanding, which is what I try to get at here, is that what a New Zealand forest looks like today, even in its pristine, fenced, non-browsed environment, is not the forest that was present 800 years ago when Māori first arrived, because of the fact that it has lost what's known as the keystone species, the moa. And so, back to colossal. So, my friend Peter Jackson has, or he learnt about colossal and said, well, this is a bloody good idea, and let's get involved, because ever since he was a child, he has wanted to see moa. And because of his success, he has a little bit of spare cash. So he has wanted, or he has effectively strong-armed Colossal to get involved in the moa project. And it's actually a brilliant piece of, well, it's fantastic because of the fact that moa actually are a very good surrogate for this particular technology. They have relatives that are of a similar size. They have a comparatively simple breeding biology. And because we have partnered with Ngāi Tahu, in particular Ngāi Tuhurere, we have a place to put them and a reason to put them there, which is tourism. As we all know, especially here in the South Island, perhaps not on a day like today, there are lots of tourists in New Zealand who come for our environment and are here to see things to do with the natural environment. They'll go to Queenstown, they'll come here and see our beautiful botanic gardens, but they're crying out for more and more ecotourism opportunities. And because of that fantastic income stream, we have the ability to provide unique biotechnology-related ecotourism opportunities, and the one that we're talking about here is an experience where New Zealanders and international tourists can see a glimpse into what perhaps you might call a Jurassic Park type experience, where you actually have the opportunity to see a mythical animal brought back to life. So in combination with the Ngāi Tahu Research Centre and Canterbury Museum, Wingnut Films and Colossal Biotechnology are working towards the idea of bringing back a functionally equivalent moa to New Zealand in the next 10 years. We know that this is possible through the technology. We've got 270 scientists working on this project, as well as the other projects, dodo, mammoth and thylacine. And we have the facilities to actually do this. We have emu in captivity in Australia. We have people working on all sorts of different parts of this project, artificial eggs, putting together germ cell technology, and literally on a daily or weekly basis, breakthroughs are being made in this area. So we have the ability to sequence the genomes even of these ancient bones. We've been collecting more moa bone from sites where the DNA has been exceptionally well preserved. And we now have a large number of complete genomes of moa. We have now got the ability, which is something that's only developed in the last 2 years, to produce what are known as primordial germ cells, which are the precursors to the ovary, eggs and sperm. Literally when this project was started, we didn't have the ability to do this. Literally 6 months ago, when I was talking to people at Colossal, they had a huge problem with this exact issue with regards to dodo and pigeons, which are the surrogate for dodo at this point in time. They thought perhaps that was insurmountable, and 3 weeks later I get an email saying they'd cracked that one. So when you put together a bunch of people like this, the very top leaders in their field, you get to solve a project or problem very simply. Now we've got just a couple of little issues with moa. The bizarre thing is that the most recent DNA study that was done led to a very unexpected conclusion that moa was most genetically related to tinamou, and yet tinamous are small chicken size or even smaller bantam sized birds from South America. Bizarrely, they can also fly, the only member of the ratite group that can actually fly. But because tinamous are small and lay quite small eggs, even though they're most genetically closely related, when we were taking a modified genes or genome and placing them in their nearest relative, the egg would be too small. So we've developed artificial egg technology, first in the world technology to actually achieve this. Just recently, we have chickens, chicken embryos and artificial eggs. But when we actually go back and look at the DNA of moa, emu and tinamou, the differences between emu and moa aren't as significant as we once thought. So it's very likely, or almost certainly now, that we'll probably use emu as a surrogate to raise the first embryos of moa. And this will solve a whole lot of issues related to egg size, and to the fact that a lot of the features of an emu and a moa, that the things that actually make it functionally a moa, like its extremely long legs, its coat of very almost hairy feathers and things, are already within the emu's genome. And the fact that we can easily raise emu either in New Zealand or in Australia, where we already have have a colony. So there's a number of technologies involved here that I won't really go into. CRISPR, which some of you may have heard, which is already being used to genetically modify people with genetic diseases, is a very important part of this project and this process. The ability to take the genome of an emu, effectively turn off all the bits that we don't want and replace them with the genes of moa to create a moa equivalent. This is the technology involved here is changing on a weekly basis. When the direwolf was actually developed at the beginning of the year before last, that we only had the ability to change parts of about 20 20 areas of the genome. We can now do tens of thousands of parts of the genome. And so, you know, literally within the last year or so, we've managed to come up with that technology. And there's an automation, ability to automate. Using robots to do some of this work. Doesn't mean that there's some exceptionally talented scientists working on this project. Last year I watched a young technician using a micropipette suck individual blood cells out of a chicken embryo. Something that apparently she was the only person in the lab who could actually do that job, so that even though there's robot technology involved here, there's actually some quite remarkably skilled people. I would certainly never have the ability to do anything like that. And there's a huge number of other steps that will lead to the eventual hatching. Surrogates will be required. And you know, this artificial smart incubator technology, this artificial egg is one of the things that we're also looking at. But why? Ultimately, there's two reasons for this. Why would we be doing this? The first is we're developing technologies that will be able to be used in conservation. At Canterbury Museum, we have more than 100 specimens of kakapo that were collected from all over the country, all from Northland down to Stewart Island, Curator. Each of those has genetic material in it which is absent from the living kakapo population today. And that information is stored at the museum in perpetuity, but without technology to be able to put that, use that genetic information to actually help the kakapo population, then we'll have nothing. We've still just got a bunch of old dead birds in a cupboard. So the ability to actually do this job, to create this technology, will have really major implications for conservation. But it'll also have implications for what we call ecological restoration or ecological healing, and an idea which is called rewilding. So I'll just take a little tangent here. If you look at North America today, the central North American plains, we have a huge number of species of plant in the prairies, the tall grass and the short grass prairies. They are, many of those are critically endangered. There's animals as well, such as the black-footed ferret. They're all critically endangered because, and one of the reasons for this is because of the fact that the environment that they currently live in isn't quite the same as it was back in the day. So back in the day, we're talking about during the era of vast buffalo herds that, you know, apparently you couldn't see the other side of as they walked across these plains. They were fundamentally altering that environment. But there were other things there. There were mammoths. There were giant rhinos. There were also giant sloths. And the exact impact of each of those different animals on the environment can only be guessed at. But what we know for sure from many experiments where some of this has been attempted to be recreated in a process called rewilding, is that the closer we get to the original pre-human environment, the more species of animal and plant will survive in an area. But then that's simply because of the fact that each of those animals and plants didn't evolve in an environment where humans were the dominant species. And for example, in the case of the tall grass prairie, a lawnmower came through twice a year to cut down the tall grass to pretend that that was the same as a bison herd going through. Because bison, of course, were bringing all sorts of other things. They were bringing huge amounts of manure. They were bringing predation. They were bringing their bodies that when they died naturally. And if we bring that analogy forward to New Zealand prior to human arrival, moa had changed the shape of the forest over millennia. Species of tree which are abundant in the forests of, say, the west coast today may not have been the species that were abundant 1,000 years ago, because they might have been species which moa preferentially like to feed on, or they may have been species that another species of extinct bird may have fed on. Moa would have spread the seeds of various species far further than just simply falling on the ground. An analogy today for the New Zealand forest is the fact that possum, the introduced possum, it very preferentially likes some species of plant and not others, and many of those species have become virtually extinct in many forests. The mistletoes of the North Island, many of those, there's a species on Waiheke Island, disappeared, and we don't really know why. It's possible that a bird that spread that species is the reason that that species disappeared. There are no possums on Waiheke. So it's our understanding of the New Zealand environment is very, I guess you'd say, in a evolving state. We understand a lot more than we did 20 or 30 years ago. But the exact role of moa and these other large or extinct birds, many of which many people haven't heard of, say Adsbill or the New Zealand giant goose, is not perfectly understood. And we have these real issues with plants and animals in New Zealand where we can't find the exact environment that they will thrive in. So it's possible that well, it's not just possible. We believe that the creation of a moa substitute in a native New Zealand environment will actually create conditions which are far more like New Zealand was 100 years ago, 800 years ago, than simply fencing off an area and turning off the lights. And we will also learn a lot about these species from this biology. I mean, in the very first instance, we'll discover their colour, we'll discover exactly what their faces look like, whether or not they were covered in feathers or not. We'll really understand their mating biology. We'll understand, whether or not, for example, they laid large numbers of eggs, whether or not they had a breeding system where the female did any work at all. Currently, we're under we believe that it's quite likely that the male did pretty much all the incubation and all the chick rearing. And so it's really quite remarkable from that point of view. The first, Peter wants a giant moa. That's what he really wants to see. But he's prepared to accept that we might have to do some of the other moa as well. And of course we do, because of the fact that to reach this goal of being closer to a New Zealand forest like it truly was 800 years ago is absolutely crucial. And Ngāi Tahu are really on board with this. I mean, and the reasons that they're on board are multiple. I mean, the opportunity to return Aotearoa to a environment which is like it was when they first arrived is a dream, but the opportunity to actually benefit from huge amounts of cutting-edge biotechnology, to actually have an economic return on some of the land that has actually been returned to them by the Crown is really exceptional. And the opportunity for employment. For example, at Hobbiton, which is another one of Peter's enterprises at Matamata, that now is employing 70 individuals. Effectively 70 families are actually being supported by that tourism venture. The opportunity for 70 Ngāi Tahu families to be supported by this venture, and also be involved actually in the science and the research. We're actively recruiting Ngāi Tahu interns to actually be involved in the science at the moment. These are really extraordinary opportunities. Peter likes to talk about this idea that we now have some of the technologies to bring back an ecosystem, a world that we believed that was lost forever. I think the quote to finish on here is this one from Peter, where he says, "As far as the health of the natural world goes, this is the most positive news any of us could ever hope to receive." And I think it's really exciting that little old Canterbury Museum is right at the thick of this and having an opportunity to be involved. So thank you very much.